Hi friends, in this video we will be talking about translation. So, what's the first thing that comes to your mind when you hear the word translation? Maybe some of you may remember like someone coming and asking you to translate something or something like that. Just imagine a stranger comes to you with a check where there is some information, maybe in Kannada, and he's asking you to translate that. So now you need to translate that to a language where that person can actually understand that, and that information will be helpful for that person. You're not imagining things and saying, the information is already over there, you're just translating it. In the same way, your DNA is having information in the form of genes, right? So the body, it can understand the language of proteins. So the language that can be understood by your body is proteins, so that needs to be translated. So DNA, so it has transcribed into mRNA, and then that mRNA is translated into the actual proteins, that is translation. So what is translation? Translation is nothing but the information which has come from DNA, it is in the form of mRNA, right? That is getting translated into the actual proteins. Or the codes on the mRNA, it is getting translated into the actual proteins. That is nothing but translation. This is the second step in the gene expression. Now what is gene expression? Gene expression is nothing but, you know that information in the DNA, that will be in the form of genes, right? So that is used in the synthesis of functional gene products like proteins. So that is nothing but gene expression, and this will take place in two steps. The first one is transcription, and the second one, as I told, translation. In transcription, what happens? The message from the DNA is copied to a RNA, that is mRNA, and this mRNA, that is nothing but messenger RNA, this brings the information outside the nucleus, and that information will be used for the second step, that is translation. Now we know that the information of the genetic code, it will be stored in the form of base pairs with the 4 bases: adenine, guanine, thymine and cytosine. In the case of RNA, in the place of thymine there will be uracil. When these bases are present on the mRNA, when that mRNA is introduced into the ribosome cannot read the individual base. It reads the bases which are in a sequence. So now I'm talking about codons. So what are codons? Codons are nothing but when 3 RNA or DNA bases are in a sequence, we call it as codons. So that ribosome can read codons, the bases which are in a sequence, not the individual bases. So this is all about gene expression. Now you know what is translation and all. Now let us come to the actual process, that is translation. How this translation takes place. So let me give you an outline, a brief idea about translation. Firstly, what happens is amino acids, when they come together, when they join, what happens? Protein molecules are formed. Firstly, amino acids should be activated. After the activation of amino acids, they are transferred onto the tRNA, transfer RNA. Then that transfer RNA comes and reads the codes which are on the mRNA. When it binds, what happens? The 2 subunits of the ribosomes, they assemble. When they assemble, what happens? That is when the protein synthesis is initiated. That's how the process begins. If you have not understood, don't worry, we'll go one by one. Firstly, the activation of amino acids. if any work has to be done, energy is required, ATP, energy currency is required, right? So firstly, imagine there is an amino acid. Now to activate this, firstly I need energy currency, that is ATP. Along with that, we need an enzyme. That enzyme is amino acid synthetase. This is the enzyme that's being used in the activation of amino acids. Then these things, when they combine, we get a complex, that is the activated amino acid. We call it as amino acid. So this is the activated amino acid. We write this as amino acid AMP with the enzyme. The complex is written like this. Plus, you know that ATP will be having 3 phosphate groups, but here you know monophosphate is there, so 2 more phosphate groups are there. So inorganic phosphates are released. So this is all about activation of amino acids. But there is one exception in the activation of amino acids. What is that exception? We'll see now. So I hope I'm clear with this. Whenever we speak about protein synthesis, the first amino acid that comes into the picture is methionine. Start codon AUG codes for that amino acid. Like a particular sequence of codons, whatever it will, a particular sequence of bases, it codes for a specific amino acid. So when methionine, when this gets activated, we call it as FMeth or formylated methionine. We call it as formylated methionine. The first codon, that is the first code AUG, the start codon is there, right? So to that, this codes for that, methionine codes for that. So we write this as FMeth, not like amino acid, that adenosine monophosphate with that enzyme. That's not the way we write this. This is one exception case. So this is all about activation of amino acid. The second step is transfer of activated amino acid to the tRNA. If you guys recall the cloverleaf model of tRNA, it looks like this: 3 arms, one short arm, 5 prime, 3 prime. 3 prime is quite elongated than the 5 prime. So this is the anticodon arm. I'm just naming the parts which I require to explain this. Here, this is the amino acid binding site. Here the functional group will be OH. So to this, our activated amino acid should come and attach. What is our complex? It was amino acid AMP enzyme, right? What happens? The activated amino acid, this amino acid binds with this. So now the tRNA is with the amino acid, activated amino acid. So it is AA tRNA. This is the complex. And this AMP and enzyme, they are set free. What is the work of this transfer RNA? Its work is to take this amino acid to the site of protein synthesis. Transfer this amino acid to the site of protein synthesis. That's what the work done by tRNA. So this is about transfer of amino acid to the tRNA. Now we have to talk about the protein synthesis. This protein synthesis will be taking place in various stages. In the first one is the assembly of ribosomes. Assembly of ribosomal subunits. You know that ribosome, it will be having two subunits, smaller subunit and larger subunit. So the smaller subunit will be having mRNA binding site, and the larger subunit, it will be having three sites: A site, P site and E site. A site is nothing but A, P and E. A site is nothing but amino acid site, or here what happens is the activated amino acid, it comes and attaches, it comes and binds there. P stands for peptidal site, there the peptide bond formation takes place, and E is the site from where the tRNA is going to take leave, or from which tRNAs are going to exit. So A stands for amino acid site, this is peptidal site, and this is exit site. So here mRNA is gonna come and it attaches here. So in the case of eukaryotes, the ribosome is of 80S type, the larger unit will be of 60S type, and the smaller one will be of 40S type. In the case of prokaryotes, it is of 70S type, the bigger one will be of 50S type, and the smaller one will be of 30S type. So where this translation will be taking place in prokaryotes. In prokaryotes, it will be taking place in cytoplasm. In the case of eukaryotes, it is in the cytoplasm as well across the membrane of endoplasmic reticulum. There the protein is synthesized and it is used inside the endoplasmic reticulum, or it will be stored there for the future usage. Now we are done with the assembly of ribosomes. No, still we have some part left. It is when these two ribosomes, when they assemble, when will they assemble? It is only at the time of protein synthesis. If protein has to be synthesized, only at that time those two subunits will come together, when they will assemble and they will help in the protein synthesis. For that assembly, we need magnesium ions. So when they are in the right concentration, in the right amount, only then they can assemble. If it is not at a required concentration, what happens? Then there will be like they will move apart. If there is like more concentration, then there will be dimer formation. So they needs to be in a required concentration. That's all about assembly of ribosomes. The next step is the attachment of mRNA. Attachment of mRNA. What happens? This is the smaller subunit of the ribosome. Here what happens? The mRNA comes and binds. This is what happens in the attachment of mRNA. It will be having mRNA binding site, right? So the mRNA comes and binds to the smaller subunit. That's what takes place here. Now we are coming to the actual part where the protein is getting synthesized. That is synthesis of polypeptide, or synthesis of sequence of amino acid chain. So now we are talking about protein synthesis. The first step is initiation. Now let me draw the diagram. The two subunits of ribosomes, they have assembled. We have mRNA, and let me write the sites. Here we have got A site, P site and E site. Now let me write the codons. the first codon that comes here is AUG. AUG is the first codon that can come here. Now what happens? Then this sequence continues, maybe U, C, C, something like that. So then there is a sequence of codons. Now what happens? tRNA, which is having the amino acid, activated amino acid, it should come and bind here. So here what happens at P side, tRNA, which is having activated methionine, that is nothing but F-met, comes and binds here because the anticodon arm can read the code here. That's what happening here. And in the A side, and this is in this P side, only AUG can come and bind because all the other codes are read at site A. Only the AUG, the first codon, the start codon, will be read at P, and rest all at site A. Now the next tRNA, here the site is still open, another tRNA comes and binds here. As I told, every sequence, it corresponds to a specific amino acid. So U, it codes for phenylalanine. So now what happened? The two tRNAs, they have come, they have attached to the code by the anticodon arm. Now what has happened? Now even the ribosome is completely assembled, everything is now done. So now what has happened? The initiation has done. When the two ribosomal subunits, when they assemble, that's when the initiation process starts. Here what has happened? The tRNA, which is having the activated amino acid, has come and it has bind to the mRNA code, which is having AUG codon. So this is all about initiation. Next step is elongation. What happens is, after this, the ribosome, it moves further on the mRNA. Like mRNA is not sliding, just leaving the ribosome. Ribosome is shifting its position. Like if this is the mRNA, ribosome is this, and this is shifting its position. That's nothing but translocation, right? So that's happening now. So when the ribosome have shifted, what happens? This AUG, it comes to the site E, right? And this UUU, it comes to the site P, and this UCC comes to the site A. And the tRNA which reads the sequence UUU is phenylalanine, and the amino acid which reads the codon UCC is serine. And we have got AUG, and you know what is this E site meant for? From here what happens? The tRNA is gonna take leave. So what has happened? From this side, tRNA will take a leave, leaving the amino acid. So the phenylalanine, this was having F-met. So when it is moving, what happens? You know, P side, it is meant for peptide bond formation. This forms a bond between these two amino acids. So formylmethionine and phenylalanine, it is in a peptide bond. It is between the peptide bond. Now what happens? This process continues. So when again this UCC comes here, then again another codon comes here. This is how the elongation takes place. You know, elongation, as the name suggests, here what should happen? The chain, the amino acid chain or the polypeptide chain, it should elongate, it should get extended, it will become longer. That's what happens here. This process continues. Then this comes here, then this comes here, then tRNA is gonna take leave, and then what happens? There will be bond formation will be between these two amino acids. So generally we can write like amino acid 1, 2 like that one also. So this is what happens in the elongation stage. This process continues till you get a stop codon that may be like UAG or UGA. So again, let me draw the smaller subunit, larger subunit, and we have got mRNA, and we know that there is FMET. There was this phenylalanine. This is P site, A site, E site, and AUG was the first one, UUU, then UCC, and then we have got UAG. We'll imagine that UAG, that's the stop codon, has come here. Now we know that UCC it was at P site, right? Now, so UCC it was at P site, UAG has come to the A site. So again, this is with its tRNA, serine. UAG, it is a stop codon. That means it codes for nothing. That's why we call it as nonsense codon. So when there is, it cannot code for anything. Like there is no tRNA to read that. There is no anticodon arm to read that. What happens? This chain is going to break. That's it. That's the protein synthesis ends there. So this is the way the ribosome gets to know that yes, my work has done. My work is finished. So now I need to take a leave. So what happens? The bond, whatever it was there, so even this comes here, and then what happens? Everything is gonna dissociate. Like ribosomal subunits, they separate. mRNA it destroys actually, and everything is gonna separate. And finally, what we get is the sequence of amino acids, that is FMET, phenylalanine, serine, and so on. So that's all about initiation, elongation, and termination. If the process has come to an end, yes, almost. Now this is not the protein which is exactly required by our body. It's not like that. So this may undergo some additional processes. Maybe there may be removal of some amino acids, or it may undergo some foldings to become more stable structure. Then it is sent to the part where that protein is required. So this is all about the translation. You know, proteins, they are very much important, as already I had mentioned that. So this is all about translation. I hope each and every concept, whatever I have discussed, it's clear.